So now moving back to the RNA world hypothesis, it suggests that RNA in the primitive world had both. It is a genetic material also that was self replicating, and it had catalytic role also. Okay, so this we have discussed in the last class about the ribozymes, and now we'll be starting with the portion of the genetic material. Okay, so now from this we'll be studying about the properties of the genetic material and how RNA is a genetic material, whether it is stable or unstable. So that we'll discuss in the following slides. So first we'll see the properties of a good genetic material and how a genetic material needs to be there. So, talking about the genetic material, it must be a molecule that can contain all the genetic information about the body in form of genes that we say, or any of these molecules that can contain the genetic information. This is the basic definition of a genetic material. So, if there is a molecule that can be called as a genetic material, so uh, like there would be some properties of that particular molecule. Okay, so first thing is that it must be capable of replication. Okay, so for example, this is the genetic material of a organism, and it if it is not able to replicate, okay, so it is not able to make its own copies, then that would not be able to pass on to the future generation. Okay, so that means ki if the genetic material is able to replicate, that means it can create its own number of copies on its own, and that will lead to passing on to the future generations after cell division. So if that is not happening, then it cannot be passed on to the future generations. That was the first property. The second one, it must be stable structurally as well as chemically. So there must be some form of structure that will be formed out of the genetic material. So, for example, this is the genetic material, and to be stable inside the cell, it would have some kind of combination with the proteins inside the cell to form a structure. For example, in our human system, nucleosome, chromosomes are formed. Okay, so this is the structure of the genetic material, and chemically in terms of, for example, nucleic acid are the form of nucleotides, so it has different groups, and it must be stable inside the system so that it cannot be degraded easily. Okay, so this was the second point, that it must be stable. Then it must have scope of slow changes or mutations to evolve. So mutations are generally referred to as small changes in the genetic material. Okay, so now these changes help evolve in evolution. Okay, so what happens? The environmental changes that we have, so that leads to evolution, and evolution is the basic need of before the mutation. So mutation helps in evolution rather. Okay, so if there is a genetic material and it is able to make small changes in itself so that the organism can evolve with time. Okay, so this is a one of the properties of the genetic material, and it is expressed in the form of Mendelian characters, that means in the form of genes. So it can express and show the phenotypic response. Okay, just for example, like Mendel used pea plant to explain the genetic characters. Okay, so he used different kinds of characters. For example, stem height. So that was a phenotype. Okay, so this phenotype was governed by one of the genes that was present on the genome of the pea plant. Okay, so this is the way that genetic material must express itself to show the phenotypic response. That means in the form of Mendelian characters, and it could pass through the generations. Okay, so like we discussed in the first point also, the genetic material must pass through generations, stably through the generations, so that it uh, basically helps in evolution, as well as it can pass on the phenotypic response that it can lead to two different generations. Okay, so these are different properties of the genetic material that must be there in the molecule that is going to function as a genetic material. So now, based on different experiments done by Griffith and the transforming principle experiment and different biochemical essays by Avery, McLeod, and McCarthy to say that DNA is the genetic material. So now moving on to RNA as the genetic material. So as we just discussed that DNA was the main genetic material because it could follow all the properties of the genetic material, so that it can replicate, it is stable, it has a scope of slow changes, that it can mutate itself, it is expressed in the form of Mendelian characters, and it can pass stably through the generations. Okay, so now RNA have some of the limitations, like it is less stable. So, and because it's less stable, it is highly reactive. Because, as you remember, this was the ribose sugar, this was the base, and here the 2' OH is also present. Okay, so O being a electron rich compound, it has a lone pair of electrons, so this is a very good nucleophile. And as you have seen in the ribozyme case also, this 2' OH behaves as a nucleophile in most of the cases where catalytic function is involved, making it highly reactive. Okay, so in form of a genetic material that must not be reactive. Okay, then it is more prone to mutation, that is, it evolves very faster as compared to DNA. Okay, so this is one of the reasons that the presence of 2' OH makes it more prone to mutation. Then, but it expresses in the Mendelian characteristics faster than DNA because, as we know, the central dogma. So, if DNA has to express itself, it must go through RNA. But if RNA is only the genetic material, then it can directly translate into proteins. So, it is much faster to express the proteins when RNA is the genetic material as compared to the DNA. Okay, so these were the properties that make RNA not so good as a genetic material, but because the RNA world hypothesis states that RNA is a genetic material in the primitive part in the primitive organisms, so this that was basically transformed to the viruses. Okay, so certain viruses have RNA as a genetic material, and these viruses that have the genetic material, they basically replicate through reverse transcription. Okay, so what is reverse transcription? This central dogma goes in one direction from DNA to protein. But after this, a scientist called Tenim, th- he basically said that RNA can also go to DNA with the help of a process called reverse transcription, so that it can stably replicate itself in the living system, and then it can integrate. This form can integrate in the living system, or it can follow the central dogma after that. Okay, so that is called as feminism. And then how what led to the scientist what led the scientists to show that RNA is the genetic material? So Franklin Conrad and Singer, these two scientists basically showed that RNA is the genetic material in the tobacco mosaic virus. Okay, so they performed certain experiments where they mismatched the proteins as well as the RNA, and they found out that the RNA is the genetic material and not the proteins. So, giving a glimpse of the tobacco mosaic virus, we'll discuss this in detail in the coming slides. So, what it has is it has a cylindrical symmetry, and where the RNA is also arranged in a cylindrical fashion. Okay, so it is a major virus that infects the plants, and that was the major model organism while in the 90s for various experiments. So now what they did was they took a common strain of tobacco mosaic virus and a masked strain. Okay, so this was the masked strain of the tobacco mosaic virus. So these were the two strains. So as per the scientist, there was a confusion that RNA was the genetic material or protein was the genetic material. So to confirm that, first what they did was they developed a technique to separate the RNA and the protein alone. Okay, so what they did was they separated the RNA and the protein, and then what they did was they mixed the RNA of one strain and the protein of the other, and then what they infected the leaf, and then they isolated the virus from that particular leaf and found out that the which type of virus was that. This was the major experimental plan that they followed. So in this case, what they did was they degraded this particular protein and only the RNA was left. So what they did was, so considering this as the common strain and this was the mass strain. So common strain would have RNA that is common and protein that was common and RNA that was masked and protein that was masked. Okay, so now what they did was different combinations. So first combination was same, protein was also common. The second combination was RNA was common, but the protein was masked. In the third combination, what they saw was RNA was masked, but protein was common. In the fourth combination, the RNA was masked and the protein was also masked. Okay, so these were the four combinations. And after this, what they did was they infected the leaf, tobacco leaf. And after this, they saw that they isolated the virus, the progeny virus. And in the first combination, they found out that the virus that was present was common type. In the second one, also the virus was common type. In the third one, the virus was masked, and the fourth one also masked. Okay, so this led to a conclusion that when the RNA was of a particular strain, this produced the viruses of that particular strain only, and not the protein component. So this led to a conclusion that RNA is the genetic material and not the proteins. Okay, so this was the first experiment conducted by the scientists to show that RNA is the genetic material in some of the viruses, taking an example of the tobacco mosaic virus. Okay, now, so as we discussed earlier also, that it is present in some of the viruses, the plant or the animal viruses. So now we'll see a bit of the classification of the RNA viruses, both plants and animals, and how they are involved in the like how they are present in that particular virus. So this is the classification of the RNA virus, animal RNA viruses. So uh, starting with so RNA can also be double stranded. Okay, one of these strand is called the plus strand and the other one is called as the minus strand. So the plus strand basically expresses itself. Okay, and the minus strand helps in replication. So some of the viruses have only the plus strand in them, some of the viruses have minus strand, some of the viruses have both strand. So that are double stranded RNA. Okay. So why I told you this? Because on the basis of this particular strand only, the different types of viruses are named. Okay. So starting with the double stranded viruses, so only one family have this particular double stranded RNA, that is reoviridae. For example, in which we have rotavirus and that causes diarrhea, and arbovirus that causes encephalitis in humans. So this has a double stranded RNA in them. Then in the single stranded RNA, we have a positive and a negative. I just showed you the difference between the both. So positive strand is present in Togoviridae, Coronaviridae, Picornaviridae, uh, Calciviridae, and Retroviridae. Okay, so out of these, the Retroviridae. has a DNA intermediate during replication. So this group of viruses can integrate into the genome. Okay. The other ones have positive strand of RNA in them, and for example, we have flavivirus. For example, the dengue virus belongs to this category, and then coronavirus is the coronavirus that also have a plus strand in their plus strand as their genome. Then the picornaviridae, we have hepatitis A virus in that case. Calciviridae have hepatitis E virus. Okay, retroviridae, HIV, human T cell leukemia virus, and all those. Then comes the negative strand viruses. Okay, so in the negative strand viruses, we have orthomyxoviridae, rhabdoviridae, bunyaviridae, erinaviridae, and paramyxoviridae. So all these like viruses have the negative strand packaged in their genome. Okay, so in the orthomyxoviridae we have influenza virus, then rhabdoviridae we have Lyssa virus, bunyaviridae we have California encephalitis virus, then Erinaviridae we have Lassa virus, Paramyxoviridae we have the common cold virus, paramyxovirus, pneumonia virus, and all those kinds of viruses. Okay, so this is the basic classification of the animal viruses based on the strand of the RNA that is packaged as their genome. Okay. So this is the like structure wise uh, distribution of the plant viruses containing RNA as their genome. So most of the plant viruses have RNA genomes. Okay, so these are the examples of different viruses. Like I have told you before, also this is the double-stranded RNA. These viruses have reoviridae, partiviridae, and varicosa virus have different shapes, but they have the double-stranded RNA in them. Then the single-stranded RNA with the negative strand, and this is the with the positive strand. So these are the examples of viruses that have a positive strand in them as their genome, and this is the single-stranded RNA which has a DNA intermediate also. Okay, but in the plant case, this is a negative strand viruses, but in the animal case, it was the positive strand virus. Okay, so nothing much to explain in this. Just to give an example that these viruses are like RNA is the most preferred RNA genome in the plant viruses. Okay, so moving on to the replication of the RNA in different types of viruses and how it is replicated in various viruses. Hmm. So now continuing with the replication of the RNA. So what happens? Sorry. So different, like I said you before, that different viruses that have different types of strands in them will have different modes of replication. Okay. So for example, when we have the positive single-stranded RNA that do not replicate via the DNA intermediate. So in that case, the plus RNA can replicate through the plus minus RNA, and in that this is the replicative form. So what it does is this is the plus RNA, and this will form a double stranded RNA. This is the replicative form, and now out of this, the plus RNA is packaged in the virion also. Okay, and it can directly encode for proteins. So this was the case with the plus RNA viruses. Now the double stranded RNA viruses, what they do was they using first transcription for the using the RNA dependent RNA polymerases, they form plus strand mRNA. So this mRNA is the intermediate for both the translation as well as the replication. So now what they do is, using the same enzyme RNA dependent RNA polymerase that have another replicase activity, they form the double stranded RNA, that is the virion, that is packaged in the viruses. Okay, and this plus RNA, that is the plus stranded RNA, then forms the protein. This the protein is encoded via translation. Okay, so this was the case with the double stranded RNA viruses. Now the negative strand single stranded RNA viruses. So what they do was, first for first we'll talk about the replication, then we'll see about the translation. So this is the negative RNA virus. So for replication, they have to first form the positive strand. Okay, so this is done by the DNA dependent RNA polymerases. Sorry, RNA dependent RNA polymerases, and this is the replicative form. And out of these, this negative strand is packaged as a virion. And talking about the expression of this particular RNA, so first this RNA is converted to positive strand RNA, then to proteins. Okay, so this is how the negative strand RNA is being replicated. Now comes the retroviruses, that is the positive strand RNA that replicates through DNA intermediate. Okay, so what happens in this case when there is plus RNA? So first it transcribes it through reverse transcriptase, that is a negative strand DNA is formed. Okay, now then what happens? This reverse transcriptase has another activity of the RNase H that will degrade this part of the RNA, and then only the one strand of the DNA is left. Followed by this, the reverse transcription on the reverse transcriptase also has a DNA dependent DNA polymerase activity. So in that case, it forms a double stranded DNA. Now, the if it has to be packaged in the virus, that is an RNA virus. So this DNA needs to be transcribed. So this transcription forms the plus strand RNA that is packaged in the virus. And this also helps in the expression of the different proteins present encoded by the genome. So this is how the single stranded RNA with the DNA intermediate is replicated through in the cell. Okay, so this was the different patterns of replication, like um, taken by different RNA molecules, different viruses that have either positive strand, negative strand, or double stranded RNA as their genome. Okay, so now what we'll do is we'll study two viruses in detail and their replication, and how they replicate their genome. So first is the human immunodeficiency virus, and the other is the tobacco mosaic virus. So human immunodeficiency virus, it basically is the member of the retroviridae, as we talked about just now, because they have positive RNA, and this positive RNA is basically replicates through a DNA intermediate. Okay, so now this virus causes AIDS, acquired immunodeficiency syndrome. Okay, so what happens in this particular case? The patient has a very compromised immune system, so that leads to various infection morbidities also because of this infection of this particular virus. Okay, and as you can see in the image also, this is a enveloped virus. It has a envelope that is basically derived from the host cell, and the capsid. So envelope is a lipid layer basically that is outside the capsid. So capsid is the protein layer that surrounds the genetic material of the virus, and this particular structure is for the HIV-1, that is a common form of virus that causes AIDS in human population. So HIV-1 targets those cells that have CD4 and CD4 as a receptor, and CCR5 or CXCR4 as a co-receptor. So CD4 is the main receptor of this virus, and these two are the co-receptors. So these receptors are basically present in the T helper cells or the microglial cells. So whenever this particular virus infects these cells, so they take over the machinery of these cells for their production, and these cells cannot perform their function. That is why if we talk about the T helper cells, so if HIV infects T helper cell, this leads to various like deficiency of the immune system, leading to AIDS. Okay, and this particular virus has a positive strand RNA that is two in number. So this is the first virus and this is the second virus. Okay, and this, as we discussed just now, that it replicates via DNA internally. Okay, so talking about its structure briefly, so in the envelope of this virus, there are two kinds of proteins. First is the GP120 protein and GP41 protein. So GP120 is the docking protein that like attaches to the receptor, and GP41 is the transmembrane protein glycoprotein that attaches the GP120. Okay, and inside the capsid there is a protein layer. Okay, so that helps in protecting the capsid. So this is the capsid molecule. Okay, and here are some of the that guards the nucleic acid or the RNA molecule. And in the RNA molecule we have two kinds of proteins. First is the integrase and the reverse transcriptase. So these two proteins helps in the replication as well as integration of the uh, particular uh, genetic material into the genome of the cell. And there are certain accessory proteins also like VIF, VPR, NEF, and P7, whose roles we'll discuss later, and some protease molecules also. Okay, these proteins are also encoded by the this particular genetic material only. Okay, so this was the structure of the HIV molecule. Now, moving on to the genome structure of what the protein it encodes and what are the different parts of the genome. So, starting with the it has a 5' LTR and a 3' LTR. So, this LTR basically helps in the replication of the viruses. Okay, the things that are involved in the LTR we'll discuss in the next slide. And there is a region that is called GAG, and it encodes the capsid proteins. So different capsid proteins are encoded by this GAG region. Then we have the Pol region that basically encodes for different polymerase. So this is the polymerase, reverse transcriptase, and integrase. Okay. Then we have the ENV, that is the envelope proteins. So this is GP120 and GP41. So these envelope proteins are also encoded by this genome only. Now we have four accessory proteins. So their function is, for VIF, it helps in budding of the virus from the cell. Then we have the VPU protein. So VPU protein help also help in the budding of basically release of the virus. Then we have the TAT protein. Sorry, it helps in transcription of this particular genome. And then we have the rep protein that helps in splicing as well as transfer of the proteins to the cytoplasm. Okay, so this was the different uh, ORFs that are present in the HIV genome and what is their function. Okay, so the main proteins are GAG, POL, and ENV, and then we have the VIF, VPU, TAT, and REF proteins. Okay, so this is the different protein structures formed by the HIV genome. Now we talk about how this particular genome is being replicated inside the cell. So, talking about the LTR. So, LTR basically stand for the long terminal repeats. Okay. So, this LTR is the region that is present or conserved in all the retroviruses, and it has three regions: U3, R, and U5. Okay, so these have different primer binding sites and all those stuff. So U3 has enhancer and promoter. Okay, so this is the site where the enhancers as well as the different transcription factors and the polymerase bind. And R is the site for the poly A and termination of the transcription. Okay, so these three help in the replication of the genome. Okay, so what happens, this is arranged in the form of this pattern. So in the 5, consider this as the 5 prime end and this is the 3 prime end. So this is the R, then U5. Okay, and this is U3 and this is R. So this is how it is arranged in the genome. And then this region is called the primer binding site. Okay, so now what happens uh, when this tRNA, this helps in primer, as a primer. So this tRNA binds to the primer binding site and helps in the formation of this particular structure. Okay, now what happens? This so now what happens? This part of the genome is converted to the DNA. Okay, so this red part is known as the DNA. And now what happens? The reverse transcriptase enzyme also has a RNase H domain. So it degrades this part that has been transcribed till now. Now, this region that has been already been reverse transcribed now attaches to this part of the genome that is the other part, that or LTR. So U3R region. So now R is already present in both the sites. So what this will act as now a primer binding site, and the reverse transcriptase now will transcribe the whole of the RNA like this. Now, with the RNase, a reverse transcriptase, it have a RNase H activity also. So it degrades the RNA, and then using this particular uh, PPT. So this is also a primer binding site. So first, what will happen? The polymerase will act up on this particular region and transcribe the LTRs first. Okay, so this is the 3 prime LTR. So this will be formed first. Now this uh, like detaches from this particular DNA strand, and then it will act as a primer for the other part. So this now forms the second strand, full strand of the uh, RNA molecule. So this forms the double stranded DNA or the provirus of the HIV genome with the 5 prime LTR and the 3 prime LTR. And this double stranded DNA is only the form that integrates into the genome. Okay, so what happens now? First, the tRNA that helps in as a primer binding site, it first transcribe the left half of the 5 prime LTR and degrades the RNA part of that particular thing that has been reverse transcribed. Then this particular part that has been reverse transcribed act as a primer for the other side. So this is called as the first jump. Okay, now this particular this particular primer will form the whole RNA and by the reverse transcription, and then the RNA part is degraded using RNase H activity. Okay, and then this is also a primer binding site for the DNA, and this will now act as a primer for the formation of the 3 prime LTR. Now this 3 prime LTR, as soon as it is formed, it will like be detached from the other strand, and it is jumped to the other side for the formation of the complete strand. Okay, now this strand is completed with the formation of the 3 prime LTR, and this strand is completed this side with the formation of the 5 prime LTR. Okay, so this is how the double stranded DNA or the intermediate form of HIV is being produced. Now, when the HIV is being packaged. So this double stranded form basically transcribes this RNA. This is the 5 prime end. So here what will we have? We have R U5 GAG Pro Pol ENV, then PPT, followed by U3 and R. Okay, so this is the 3 prime. So now this will lead to the first step, and this is how it is packaged. Okay, so this was the replication of the RNA strand in the retrovirus or the HIV that we were talking about. So this comes the end of the HIV wala part. Now we'll move on to the tobacco mosaic virus. So as we just discussed that it is a cylindrical virus, and it is basically formed of different proteins called as capsomers, and the RNA is also arranged in the form of a cylinder. So it has a hollow core. Okay, so like this you can see. So the RNA is attached to the capsomers, and it is like arranged in a helical format, and it has a hollow core, and the radius of this core is around 40 Armstrong, and the pitch of this particular capsomers is around 23 Armstrong, and the whole diameter of this particular tobacco mosaic virus is 180 Armstrong. Okay, so it basically infects the tobacco plant and different other plants also. Okay, so this is the genome organization of the basic TMV virus. So what happens? This has a genome in which this is a long protein followed by some subgenomic promoters that leads to the and like formation of different proteins and have a tRNA like structure, and it has a 5 prime cap like present in the eukaryotic system. So first, when it transcribes, it forms a replicase protein. This is a replicase protein, and this like this is a leaky stop codon. So this is a so due to this leaky stop codon, it forms a protein that is RNA dependent RNA polymerase. Okay, so followed by the subgenomic promoters, it forms one is the movement protein and one is the coat protein. Okay, so this is the capsomers. It forms the capsomers, and this movement proteins helps in the movement of the virus through the plant body following the vascular path. Okay, and this tRNA like structure help in formation of the second loop or the second strand of this particular virus, so that it will be that will form the replicative form. Okay, so how it replicates that we'll see now. So it is also a plus RNA strand virus. Okay, so now based depending upon the RNA dependent RNA polymerase, it forms the second strand, that is a replicative form. And now there are multiple negative strands RNA as formed, and based on that, one will so using that various positive strand RNA are formed. Okay, and this positive strand RNA are used both for the packaging into the virion as well as into the translation of the movement proteins and the capsid proteins, or as well as the replication proteins for replication. Okay, so this was the basic idea about the replication of the tobacco mosaic virus and how it gets like how the different proteins are encoded in the human immunodeficiency virus and the tobacco mosaic virus. So this comes to the end of the lecture that RNA can act as a that RNA, although it is reactive, less stable, but it can act as a genetic material in some of the viruses, the plant or animal viruses, but not of the other organisms. Okay, so this was the whole idea about the genetic material, and from the next class, we'll be starting some new topics. So stay tuned and thank you for joining.